Sure. So we are using regulatory T cells, and I'm sure you are aware of the basics of the biology of those cells. They are used to control down the immune system when the immune system has turned against the self. And what we are using is that we are using CAR technology, which is well known and well described in oncology. In oncology, you are using a CAR technology in what we call a T effector cells. But in our case, we are using the CAR technology into a Treg. So very logically or very naturally, we are using those CAR-Treg not to fight cancer or infectious disease, but to fight autoimmune condition, inflammatory disorder, or transplantation-related disorder. And we believe to be the only player today in the world to be focusing exclusively on the usage of the CAR technology into regulatory T cells. You basically have two other Treg development that we know of. One is the use of what we call polyclonal Treg in autologous cell therapy processes. And the other one, which aim at using ways to stimulate Tregs in vivo, for example, with low-dose IL-2. We have a different technology. We use Tregs extracted from patients, but we do not re-inject them as they were after amplification, but we genetically modify them to make them antigen-specific. That's the main difference with the usage of polyclonal Treg in other cell therapy development. With respect to the technology that aim at stimulating Tregs in vivo, well, obviously this is a different approach. We come with something which we believe is much more personalized and also much more targeted. Personalized because it's autologous, so from patient back to the patient, and targeted since, again, we introduce a CAR. So again, you are talking about a different kind of technology. We believe to have, again, something which is totally unique, more personalized and more targeted. Sure. So this technology has never been tested in man, and so we wanted to go into a first-in-man study in 2018 with a product which is more, let's say, acceptable for the authorities as well as for the patient. So we moved from a murine SCFV, so antibody fragment that is used in the CAR, to a humanized version of the SCFV. And the expectation is that with this humanized version of the CAR, we will have a better tolerance profile, and also we will probably avoid the immune system of the patient to fight against the CAR-Treg that we will inject back to them. Well, the question is not too much could we use it multiple times, is will we have to use it multiple times? And only the initial clinical development plan will be able to answer to that question, and there is basically two outcomes. We have to inject patients multiple times over time, so we have a treatment which becomes chronic for a chronic disease, or we change or modify the disease, we cure the patient, and then we will have to inject the patient only once, but it's too early to say. Of course, the second outcome will be much more preferable for the patient as well as for the company, but it's too early to say. Our first project, the one for which we expect to start clinical development in 2018, is in solid organ transplantation. Next in line, we have like 5 or 6 different programs at different stage of preclinical development. Probably the most advanced of the other program is in multiple sclerosis, and I hope that soon in the near future we'll be able to give some more updates. And what exactly do we have and what will be the timeline for development of this candidate. But it's, again, too early to say today. Sure, so we have recently announced that we've secured a bridge facility should the warrants, the cash warrants that are listed and that will expire at the end of February 2018, not be fully exercised. So this bridge facility is theoretically enough for us to go through 2018 without any issue.